And okay, so I'm gonna start. It's a great pleasure to introduce Matthias Meier to you. We are very close friends. I would even consider him as more as family than just a friend. And so Matthias went to high school in Burghausen, it's in Bavaria, and studied biology. And we met the first day of immatriculation, and then from then on we were inseparable. We were even called ETMM. Exactly. So Matthias did his bachelor's with honors for 4 years at the LMU in Munich, and afterwards joined Axel Borst's lab at the MPI of neurobiology. He did his PhD there and is now also performing his postdoc research. It is like his publication record, I would say, is exceptional. Already during his PhD, he has published 6 high-impact papers, and now is still continuing on producing excellent research every year with, I think, a lot of new findings that he will present in today's keynote lecture. So Matthias, I'm happy to have you here, that you give us some insight about Drosophila motion vision, and you can present, and I'm happy to listen to your talk.
Thanks a lot, E.T. First of all, thanks for the very nice introduction. I have to say that...
Yes, I do not know why it's not working.
Can everybody hear me?
Yes.
Okay, fine. So the first thing I wanna say is that all of these 6 high-impact publications I had during my PhD were in collaboration with Etienne. So it was a very special kind of group that we had in the lab, and that was also very fruitful and very productive. And actually, at least I tried to build up my presentation in a way that I kind of tell the story of when I entered the lab and what the kind of current status of knowledge was back then, and then what happened during my time and our time in the lab, and what kind of is going on at the moment. And also, one thing I want to say before I start is that I really enjoy kind of interactive presentations. I'm not sure how well that is going to be possible today, since we're not all at the same place. But I think ET is going to take care of the questions, because I only have one screen, so I cannot see my presentation in your faces at the same time. But if you have a question, so either if you're present, just tell ET and he's going to interrupt, or raise your hand, or say something in the chat, or whatever. We're gonna try to make it as interactive as possible. Okay, then I'm gonna start my screen sharing. 1 minute. Okay. Let me start the presentation. All right, so I have to be honest, I was struggling a tiny little bit with putting the research into the framework of the tiny machine learning. And actually the only machine learning part is going to be slightly hidden in the title, with learning from tiny machines. But in general, I'm going to talk about motion detection in fruit flies or in Drosophila. Okay, let's get started. So why is motion detection even relevant? I'm not sure if any of you remember this movie. It's Jurassic Park, and here what they say is that the dinosaur can only see you when you move. So with this, I want to claim that motion detection is kind of survival critical. But more seriously, oh wait, I'm gonna put on a laser pointer. Okay. I have a few samples of simple visual behaviors that are actually based on extracting visual information or motion information from a visual scene. The first one here on the left is the so-called optokinetic reflex, and I'm sure all of you actually know it. Because if you, for example, if you're sitting in a car or in a train and you're driving by a visual scene, then your eyes will always make these small movements that you can see here. And this is actually already a computation that takes place in your eyes, in your retinas, that calculate the direction of motion and will then initiate a response. And something quite similar can be also seen in other animals. So this is a so-called zebrafish. It's a zebrafish larva that is embedded in agarose, so the head is actually fixed, and then the tail can move freely. And underneath this fish, they will show a visual stimulus, and you can see that when the stimulus moves to one side, the tail of the fish will move to this one side. And when the direction of the visual motion switches, the fish will respond in the opposite direction, trying to stabilize its swimming behavior. And here on the right, I have an example of moving flies. So what you see here, and this is a kind of a visual arena, and when I will play the movie, you will be able to appreciate that the surrounding is going to turn in one direction. And then there's some tracking algorithm that actually tracks the path of the fly, and you can see that it nicely follows this motion of the visual surrounding. And this is what we call in the flies, it's called the Optomotor response, or the Optomotor reflex. And this is actually the basis of our research. This has been already shown a long time ago, and this is what we tried to figure out in the BOST lab at the Max Planck Institute in Munich, is how this reflex is kind of calculated by the brain, or how the visual information is processed so that the flies can respond in this way. Okay, so I want to start with a little bit of history on the subject. There was in the 1950s, there were two quite famous German researchers that invented the so-called Spangenglobus, which is one of my favorite kind of German science words. And what it means, it's kind of like I looked up the word for Spange, but I forgot again. But it's what you see here. So they took this little beetle or a weevil, Chlorophanus viridis, and they put it on actually bent and glued straws, and they made them kind of not walk on them, but move the straw with their little legs. And then it was shaped in a way that every now and then, or every kind of quarter turn, they were put into a decision point where they had to go either left or right. And then they put this whole beetle with the Spangenglobus into a visual arena. And they would show these stimuli where they had like a striped pattern, for example, that they would move either left or either right. And then they would sit there and write down the decisions or the turning points of the little beetle. And actually, I like this experiment because it's also kind of backyard brainsy, so it also fits to this for this talk. And actually, what they found out is that these beetles follow this optomotor response, and based on this and more careful research, they came up with a very famous model, which is the so-called Hastenstein Reichert detector. It's an elementary motion detector, and I will slowly walk you through this. And it's kind of a theoretical model that would explain. So in the beginning, it would explain their findings about the turning behavior of the beetle. But as it turns out, it's also a good model for the neuronal responses to visual motion. And it consists of basically two arms. So up here, you have the input regions of the detector. So you have two spatially offset inputs. And then you have down here a nonlinear interaction stage. So let's keep it very simple. Let's say here in the Reichert detector, the signal gets multiplied. And importantly, these two arms of the detector, they're tuned differently. One of them has a temporal delay, and the other one is fast. So let's move a visual stimulus from left to right, which is the preferred direction of this detector, and let's see what happens. We can see that the delayed arm delays the signal, and they will coincide at the interaction stage and result in a strong signal. When the signal moves into the opposite direction, the so-called null direction, they will not coincide and they will not get multiplied, so you have a weaker signal. And this actually leaves you with a direction selective signal with a very simple model. A few years later, also Barlow and Levick came up with a similar model, actually based on neuronal data. And it looks like this. It's very similar, but the delayed arm is on the other side. And importantly, it has a inhibitory synapse. So that means that the direct signal gets inhibited in null direction. Let's see what that looks like. Sorry, that's a weird GIF. So the blue one here is now the inhibitory signal, and they do not cancel out in preferred direction. So you get a signal in preferred direction, but in null direction, as you will see down here, the two signals will cancel out and you get very little response. And now I'm gonna kind of skip quite a lot of steps actually, and I will tell you that what us and many other researchers have figured out now, that in the fruit fly, there's actually a combination of these two models that can be found. So when we look at direction selective signals in the flies, and I will come to that in a second, we can actually find both these enhanced signals in preferred direction and these reduced signals in null direction. Okay, are there any questions so far to this theoretical part? If not, then, so you have to speak up because I don't see you. If not, then I will move on a little bit, and I will very, very briefly mention the vertebrate systems, where you can find direction selectivity actually at several stages in the brain, already at the LGN and then in the visual cortex back here, that I know some of you are working with, I guess, for the projects. And that's a super, I think one of the most famous neuroscience experiments by Hubel and Wiesel, who found in the cat visual cortex, found these direction selective neurons where they would show these bars that move in different directions, like either left to right or right to left, or they would turn it and then they would move more in a 45-degree angle or upwards downwards, and they would find cells that only respond when they are stimulated in a certain direction. So these neurons are direction selective. And quite a bit later, so this is from a review, that's not the original work, but it could be shown that also already neurons in the eye, so in the retina, are direction selective, which is pretty cool because that means that the eye already does quite some computations on its own. So there is a lot of math going on already in the retina and not just in the visual cortex. So what we see here, for example, is a neuron that will respond very strongly when stimulated in preferred direction and will not spike at all when stimulated in null direction. All right, but now let's move to our object of studies. This is the Fruit Fly Drosophila. And you can see they have slightly different eyes than we do, because they have these compound eyes with like hundreds of different facets. I think recently somebody counted them. I think it's about 700 in total, or 750 or so. And behind these facets lies the brain of the fly, and actually behind the eyes are the two so-called optic lobes. So you can see that actually the fly devotes almost half of its brain to the processing of visual information. So these two lobes are really there to process visual information. So the flies are a highly visual animal, which is also one of the reasons why it's very interesting for studying. There are several others that I will come to in a second. All right. Again, I want to start slightly historically. There was this study by Carl Friedrich Fischbach and Dittrich. I guess the study was made over the whole 1980s or even before, and it was published in 89. And what they did was do Golgi stainings to look at almost all of the neurons of the optic lobes of the fruit fly. So very quickly, Golgi stainings were developed in the 19th century by Golgi. It's kind of a silver staining where somehow miraculously only some neurons are stained. Still not quite clear how that works, that only a few neurons get stained, but the output of that is that you can actually look at the anatomy of single neurons. Because if you were to stain all of the neurons in the optic lobe, then you would only see one big black blurb in this case. But with this Golgi method, you actually get single cell clones of the neurons. So this Fischbach guy, he actually took really many years, did these Golgi stainings of the fly brain, and then drew a huge atlas for all the different cell types he could find. And it's hundreds of cell types that he very precisely drew. And this is really, I think, the basis for our field. And this is like every time we look at a staining or whatever, we try to figure out, okay, what's that neuron type? And then we open Fischbach and Dittrich and we check it out. So this is really a very important work for our field. All right, so that's kind of the basis. But now the question is, how is this model that I briefly talked about in the beginning, how is that implemented in this jungle of neurons in the optic lobes of the fly? So the neural implementation of direction selectivity has often been referred to as the holy grail of motion vision in Drosophila, especially by our boss. Okay, so how do we tackle this problem in Drosophila? Because for a very long time, now this is slowly changing, but for a very long time, most of these cells were not accessible because they are actually quite small. So the brain of a fly is about 1 millimeter large, and these neurons, the somas, they are 1 to 2, like at least for these neurons, they are 1 to 2 microns in diameter. So they're very tiny, and it has historically been very difficult to record from them with electrophysiological methods. So meaning either with sharp electrodes or with patch clamp, or just ephys is hard in Drosophila in general. But there are some neurons that are different. So here I want to quickly introduce the kind of overview or the general Bauplan of the optic lobes. So what you can see here, so the light enters here from the left. This is the red structure is the retina. Below that you have the medulla, the lamina, the medulla, and the lobular complex, the lobular and lobular ped. It's actually, you don't have to remember that. Actually, that's useless information for this talk. It's just so in the system that we always do this introduction, but it's actually not important. What's important is that you have these large neurons down here at kind of the output of the optic lobes that are direction selective, and they actually have big somata, so it's fairly easy to record from them. And exactly, the important thing from this slide is that when you were to record from photoreceptors that are sitting here in the first layer, you would see, independent of the direction you would show, the direction of motion you would show, you get the same response. So these cells here in the retina, they just respond to changing luminance. So does anything get bright or dark? They will realize that, but they don't know in which direction to move. A few synapses downstream in these large tangential cells changes, you get direction selective neurons. And this is actually what they look like when recorded from a soma of these neurons. So somewhere in between the input and these tangential cells, the direction selectivity must be computed. And that has been for a long time kind of the quest. How is that possible? And actually, at the time when Etienne and I joined the lab, it was still quite unclear. So this is when we joined the lab. We knew that there are these tangential cells, they are direction selective, but we had these Fischbach-Dittrich Golgi stainings of all the neurons, and kind of the job was to figure out how this computation is done. Of course, I'm only talking about our lab now. There are also other labs that worked on the same problem and that were also very productive. But since this is kind of I'm mostly going to talk about our lab, but at the points where it's important, I also reference the work of other groups. Okay, so the huge advantage of Drosophila melanogaster as a model organism is its genetic accessibility. So there's really so many different tools that you can use and that are especially very, very easy to use because there are other groups that develop them. There are other groups that actually create these fly lines. So all we have to do as kind of the end user scientists is we take a fly line and cross it to another one, which is putting them into a vial and waiting for a week. And then the new flies hatch, and then we can do our experiments. So it's really very, very handy. And I want to briefly introduce two genetic tools that we use a lot in the lab, or that we used a lot in the lab. The first one is Shibiri, and that's actually a genetic blocker. So what it does is that so the cool thing about Shibiri is that it's temperature sensitive. So that means if you're at room temperature, let's say 25 degrees Celsius, I have no idea what that is in Fahrenheit, the neuronal transmission, the synaptic transmission would work regularly. But then if you move the temperature to 30 degrees, something in this protein called dynamin, which is important for the reuptake of vesicles in the synaptic cleft, also details not very important, breaks down, and the synaptic transmission does not work anymore. And what this does to the cell is that it kind of silences the neuron, so it doesn't talk to its postsynaptic partners anymore. And that's pretty handy for us actually, because now what we can do is we can, for example, use the patch clamp technique, so record electrophysiological signals from these large tangential cells down here, and at the same time take out neurons upstream and see what happens. And the second tool that's really important and that only just kind of arrived at the time when Etienne and I joined the lab is calcium imaging. And that was really a revolution, because what you can do now is using light microscopy to look at neuronal activity kind of live. And it doesn't matter how tiny the neuron is. You can see the signal. You don't need an electrode to record from it. I mean, there are downsides of it, but it really made our life a lot easier at the time. All right, let's see what the next slide is. Yeah, so with this huge kind of toolbox in Drosophila, as I said, there are thousands of different fly lines. There's a website where you can literally order a fly line that has expression in a certain cell type. You go there, you put it into your shopping cart, and you pay, and then you get the fly line delivered. So it's really very, very kind of handy science. Okay, so now let's get to see some data. So this is the work of 2 PhD students that were in the lab when I joined in 2010 and then left, I think, in 2011 or so. That's Max Hirsch and Bettina Schnell. They're both group leaders now. Max is in Vienna and Bettina, I think she's in Bonn. But correct me if I'm wrong, Bettina. And so I'm going to slowly walk you through these graphs. So what they did was they used these patch clamp recordings from lobular plate tangential cells and then visually stimulated the neurons. And so let's first only look at this black trace. So they showed an on edge, which means that they have a dark screen and then it becomes bright from one side in the preferred direction of this lobular plate tangential cell, which let's say is from left to right. In the preferred direction, so it depolarizes, so there's an increase in activity. And when they move it from right to left, so in the null direction, there's a decrease in activity. And a decrease. So there's an inhibition. And the same is true when they used off edges, so dark edges. So again, hyperpolarization in null direction, depolarization in preferred direction. And then what they did was they expressed this Shibiri I just talked about in only these L1 cells. It's not very important where they are, but they're kind of at the input of they're just behind the photoreceptors. So they get signals from the light-sensitive neurons. And when they did that, when they blocked this L1 cell, then the responses only to the on edges were gone, but the responses to the off edges were still there. And they did the same experiments with blocking L2, and they found the opposite. So on responses were still there, but off responses were gone. And that was a pretty cool finding, actually, because this has also been known in the vertebrate retina, that there are neurons that only respond to brightness increments or only to brightness decrements. So there are on-specific and off-specific cells in the vertebrate retina, and as these two and also other colleagues in the lab, other groups found out, is that the split also happens actually in Drosophila, which is pretty cool. So now that was established, kind of the big hunt started in the field for which are actually the first direction selective neurons. Because these, as we just saw, they are not, no, actually we didn't see it, but there was another study that also did calcium imaging from these neurons, but they're not direction selective. So they respond irrespective of the direction they're stimulated. But there were a few quite promising candidates for direction selective neurons. And there were two candidates. They are the so-called T4 and T5 cells. So I think these are the only neurons that are worth remembering now in this talk, because they are pretty cool, actually. So these T4 neurons, they connect the medulla, so this part of the fly visual system. Sorry, I didn't introduce this here. So what we see up here is kind of a schematic representation of the visual, of the optic lobe of the fly. So the input is up here, and then these tangential cells I talked about before that are direction selective, they are down here. And so why were these T4 and T5 neurons good candidates? So first of all, they exist in 4 subtypes. And again, something I didn't tell you, but I'm telling you now, is that this lobular plate and these lobular plate tangential cells actually has 4 different layers. And the cool thing, and that had been shown before with like radioactive labeling techniques, is that these 4 layers, they respond to the 4 cardinal directions of visual motion. So that means upwards, downwards, leftwards, rightwards. And the cool thing now is that these T4 and T5 neurons, they exist in 4 different flavors. So they exist in one subtype for each of these layers. And the T4 cells, they connect this neural pill to the lobular plate, and the T5 cells, they connect this neural pill. So they were actually quite strong candidates already. And now, I want to quickly introduce you to what actually the experiments look like in our lab. So this, I like this picture because there's this Backyard Brains cartoon where they show kind of expensive scientific equipment and how it's unnecessary and how you can do everything with a little box. And I find that this perfectly looks like the cartoon, like a very complicated box with many different cables and lights. And this is actually the microscope we're using for recording the activity with calcium imaging in the brain. So here you see the objective. The fly is actually under this objective, and what's shielded under this black foil is the visual kind of, we call it the arena that stimulates the fly visually. That's kind of it's a screen, but a round one. All right, and now, so this is what it looks like when we take a look at the activity of one of these. So actually now what we have here is a T5 neuron. So in this panel here, you will see the activity of this calcium indicator when I stimulate the fly visually with a pattern that moves in 2 different directions. In this case, it moves up and it moves down. And what I did here is that I expressed this calcium indicator only in one subtype of T5 cells that is sensitive to upward motion. So you will see that the cell only actually responds when the stimulus moves up. And I really love this experiment because it's so obvious, and you get such a strong signal. And you see when the stimulus moves down, then there's no response. I will show it again because I really like it. And now actually there's also an edge. But maybe I was... I'm sorry, yeah. Okay, exactly. So it's a T4 neuron, actually, not a T5. But I didn't tell you the difference yet, so it doesn't make a difference for you. Okay, let's move on. So in 2013, or actually before, but let's say in 2012 probably, kind of the technique of calcium imaging was being properly installed in the lab, and people did all of these experiments with T4 and T5 cells. And as the example I just showed you, here we have both T4 and T5 of all 4 subtypes. And it's a bit hard to see here, but when you stimulate them in these 4 cardinal directions, right, left, up, down, what you can see is that these 4 different layers actually respond only to one direction at a time. So that was really a pretty cool result at the time, that we kind of could show, or there was experiments done by Matt Meier and Jürgen Haag. Matt was a PhD student at the time in the lab, and Jürgen is kind of the long-term postdoc, or kind of a scientist in the lab, who's really, I think, the most important person in the lab, because he knows how experiments are done properly, and he knows how the microscopes work. And if there's a problem, we would always ask Jürgen, and he would tell you within 2 seconds what the solution to that problem is. So, but what they did was they were able to show for the first time that actually these T4 and T5 neurons are direction selective. So they respond only to one direction of visual motion. If I'm being too slow or too fast, please let me know, okay? But the other cool thing, what they could find, is when they presented them not with moving gratings, but again with these moving edges, is that the neurons there are also polarity specific. So that means that T4 cells only respond to on edges, as you can see down here in these bar graphs. So always the bright one is an on edge and the dark one is an off edge. but they don't respond to off edges. And the T5 neurons only respond to off edges, but not to on. So kind of the split in on and off that I was talking about in the beginning still exists at the level of these T4 and T5 cells. So they're direction selective, and they're also polarity specific. So that was pretty cool. At the time they had submitted the paper to Nature, they said, yeah, cool, but somehow not interesting enough. So if you want to have a chance in Nature, we also want to see whether these cells are actually necessary for direction selectivity. And that's how Etienne, me, and another colleague slipped onto this paper, because we did electrophysiological recordings. So we kind of repeated the experiments that I talked about before for Max and Bettina, where we recorded from these large lobular plate tangential neurons and blocked the output of T4 and T5 neurons separately. So here on the left panel, we see controls, 3 different controls, and in the upper 2 panels, there are on edges, and in the lower 2 panels, there are off edges. And on the right here, we can see when we block T5 cells, so the orange line here, then the response of these lobular plate tangential cells to on edges is completely unaltered. However, when we block T4 neurons, then there's almost no more response to on edges. And the other way around, when we showed dark edges, blocking T5 cells almost completely abolishes the response of lobular plate tangential cells to dark edges, but blocking T4 cells leaves these responses intact. So with this, we could actually, there was a happy end for this paper, and we made it into Nature. And the reason why I showed this is not to brag, but because the really cool thing about this study was that it was super collaborative. So almost all of these people that are here are actually from the lab, were PhD students at the time, with a few exceptions that were postdocs. But it's really, I think half of the lab is actually on this paper, and that's actually why I really like it, because it made everybody happy and was a great success story. Okay, so where am I time-wise? I've been talking a lot. So in the meantime, that was 2013. A lot has happened. So there were many more studies being published on what is in between kind of these direction selective neurons and the input neurons. And I'm not going to go into detail about this. Just very briefly, we could find that, so here in this middle panel, Etienne and I could find that in the off pathway, there are actually at least 4 neuron types that are important for computing direction selectivity in T5 cells. At the same time, Georg Ammer, a colleague, showed similar things for the on pathway. And then there were other colleagues from our lab that, again, characterized all of these kind of in-between neurons, I'm going to call them, very carefully, and could show their response properties and their filter properties. And here I also want to briefly mention all the other labs that work on similar things and that also contributed very important findings. Okay, so I don't know, time-wise, I can either stop here or I can tell another small side story that is more recent.
No, the Ct1 story is quite important. Please tell us the story.
Okay, I will tell the Ct1 story. So the Ct1 story goes like this. That's actually, there's this super awesome neuron in the Drosophila brain. And this is actually a electromicroscopic reconstruction that a colleague of mine, Amalia, in the lab did from data that is published. So one cool thing about Drosophila now is that there's heaps of EM data that is on the internet that you can just download and then you can trace it. And the cool thing about this neuron, so what you can see here are 2 neurons. So one that has its soma here, and then it goes to the right hemisphere, and the other neuron does the same for the left hemisphere. But they're huge. They really innovate kind of the whole visual scene in the medulla and in the lobula. And it was kind of always a big puzzle what this neuron does. So the kind of obvious idea was that, yeah, it's probably somehow regulatory. It, I don't know, secretes some neuromodulators, or nobody really knew. But kind of as a side project, I think somebody already looked a bit at the neuron, but the expression was a bit weak, and then let it go. And then at some point, I think I didn't have that many experiments planned, so I thought, okay, I'm going to have a look at this neuron again. And also, exactly, one very important thing is that another EM study showed that this neuron seems to be important in the circuit for motion detection. So it seems to play a role because it has quite a lot of synapses with both T5 and T4, and that's why we thought, okay, let's just have a look at it, what it does. And so here's another quick overview of what it looks like. So it actually has its soma here, and then it has like hundreds of tiny, I call them dendroaxons, because they're neither dendrites nor axons, both in the lobula, so this is where the T5 cells are, and in the medulla, where the T4 cells are. And this is a confocal stack of the same neuron. And you can see that it actually has these tiny little compartments. And it actually perfectly matches with the retinotopic structure of the fly brain. So again, if you imagine the compound eyes of the fly, I think that's also something I didn't mention in the beginning, but it's also only important now. This kind of compound or retinotopic structure goes all the way down to the lobular plate. So you will find kind of these ommatidia, or these, I forgot the name. Yeah, kind of these these compartments all the way down to the lobular plate. And these, the Ct1 dendroaxons or exodendrites, they actually tile the whole visual field. And this is what this looks like under the 2-photon. So under the microscope that we use for doing calcium imaging. You can see here these little compartments in the lobula and the compartments in the medulla. And yeah, so in this study, I looked at calcium imaging data, and the cool thing is that our boss is actually still quite active in doing actual research. So that's one thing that's cool about the Max Planck. The Max Planck landscape is that he doesn't have to spend a lot of time on writing grants and stuff, so he can really focus on still doing research, and he does compartmental modeling. Actually, he rediscovered that with this study. So what that means is that you kind of mathematically model how the voltage spreads through a 3D reconstruction of a neuron. And so we had this 3D reconstruction from EM data, and Axel, our boss, he modeled the data. And actually, one thing that was really cool when we looked at this EM data is that, so what we see here in different colors are actually the different compartments. So the different columns in the lobula and the medulla. And actually, one column then is connected to the next one with these crazy loops. So we were super curious, why are there these weird loops? So what do they do? And what I did then was to do calcium imaging. I wanted to put a slide that explains reverse correlation. So basically what we do is I record a movie of the signal, and I show a random noise pattern. So it's basically stripes that are, I don't know, 2 degrees wide or 5 degrees wide, I don't remember, and they change brightness randomly. So they become brighter or darker, and it kind of looks like it flickers around. And then what you can do is you can take a look at the movie that you recorded of your activity, and you can back correlate your activity to what the stimulus looked like at the time the activity was high, for example, in time and space. So what you get is kind of these representations where, let's say, when let's pick one of these ROIs. So ROIs is a region of interest where I kind of average my data, and I look at this, and I can say that this one terminal actually has quite a small receptive field of, like, let's say 10 degrees of width or a bit more, and it looks at a certain point in space. And here, actually, this retinotopy, so that one point looks at a certain point in space, or one dendrite, is quite nicely shown here, because you can see that, for example, this compartment looks at this point in space. So on the x-axis is the azimuth, so the kind of this axis of visual space, and the y-axis is the response. And you can see that they really properly tile up the space, and they do so in the medulla. They respond positively to brightness increments. So this one neuron is kind of on selective in one space, and it's off selective, so it responds to negative changes in luminance, so the dark stimuli just next to it, so in the lobula. So that was kind of the first finding, was to see that there's this one neuron that actually can have two different response properties depending on where in the fly brain it's located. And the second thing that we kind of concluded from that is that one compartment actually only receives information from one point in space, so it actually kind of works like one neuron. So the cool thing about this cell seems to be that it's just one cell, but it acts like many hundreds or thousands of different neurons. And then, as I said, we wanted to verify this with this compartmental modeling, and that's the work of Axel here. And let's take a look at panel A here. What you can see here is actually the voltage distribution when you inject a certain current into a certain position. And the position is always indicated by this electrode here. So if we inject a certain current into this one compartment, then the voltage only spreads in this one compartment, and only very, very little goes to the neighboring compartments. So they are actually electronically compartmentalized, these neurons. So kind of the take home from this is that you have one huge ass neuron that actually acts as many independent processing units, which was quite a bit surprising to us. And yeah, this is what I said in the beginning. Why is that relevant for motion detection? And it is because actually, exactly, if you remember the detector I talked about in the beginning, and you remember that I said that it's this combination between this Reichert detector and the Barlow-Levick detector, so this preferred direction enhancement and direction suppression. This Ct1 neuron is the only inhibitory input that we can find on the dendrite of T5 cells. So we actually need this neuron for our model to work. And so it's kind of cool that we could show that it actually can work. And at the moment, I'm doing experiments to show whether it's actually involved and whether it's actually important for the calculation. So again, I'm trying to knock out the neuron or to silence the neuron, which is a bit tricky in this case, but I think it's going to work. And to show that it's actually implicated in the circuit. All right. So with this, I want to briefly show the people from my lab. So here you can see that's actually Axel, our boss, who who runs the lab and who's really a great guy and who gives us a lot of freedom and also a lot of supervision. So yeah, I think totally he's really a boss. Yeah, I wished to have if I hadn't had him as a boss. And here's E.T. And I've seen also Alyosha is in the audience. He's this guy here. All right. So thanks a lot for your attention. And now I'm gonna get out of sharing mode, and then I can see you again. Okay, thanks. That's it.
Perfect. Thanks, Matthias.
Okay, wait now.
So thanks a lot for the talk. I think Alyosha raised a hand, right?
What?
You just wanted to show that you were on the picture.
I was just trying to tell Matthias that I'm here. But I actually have a question. I'm
happy if there's anything you didn't understand, Alyosha, I'm happy to clarify.
No, this was a great talk. Thank you. I have one question because I genuinely don't remember. So what's the delay roughly between motion onset and LPTCs responding to the stimulus? And I'm asking about this because it's a really big machine learning problem that the latency is often so high for these networks. So I saw a talk by a guy from Tesla talking about their networks for self-driving and all this stuff. And one issue that they deal with a lot is bringing down the latency to a point where the car can respond in time with networks. Do you remember roughly how long it takes for an LPTC to respond?
It's very few milliseconds.
Yeah?
I don't remember. I don't know.
Okay, fair enough.
I think it's a few milliseconds. But I don't remember, to be honest. I don't know.
But it should be crazy fast, right?
It's very fast. Very fast.
That's cool.
Mm-hmm. Can I ask a question?
Sure.
So I used to work in zebrafish, embedded zebrafish. This was a great reminder of good science. Could you say something about what the motion selectivity is used for? So you mentioned the orienting movements that the flies do when the world is spinning, and the zebrafish do the same thing. But I've always had a hard time understanding what this motion information is actually for. So is it just to orient yourself so that the world can stay still, so that you can then apply kind of the typical methods that we use in deep learning today, where you have a still image? Or is there something more going on because it's hard to even imagine what the world would look like if you just remove the motion selectivity? Do you know what I mean? Because you could still see a still image, but you wouldn't see the moving thing. It'd be like Jurassic Park, like you said. What do you think how is it used further downstream?
It's actually super funny because it used to be at least the classic introduction for talks and theses and papers in our field, saying that a fly wants to fly through the air and is kind of brought off course by a gust of wind, and then it uses this motion information to remain on course. But this actually, until very recently, hasn't really been shown that this kind of motion information is actually used for course stabilization. Mm-hmm. But actually, Alyosha recently co-published a paper on that, where they could show that actually motion information is important for course stabilization. Mm-hmm. I mean, there are other also behaviors where the motion information is used for, but that doesn't necessarily go through the same pathway. But there's, for example, like object detection, where also motion information gets in, or this looming. For example, this looming information is very important. Mm-hmm. I don't know if you know about these giant fiber or these escape behaviors, for example. So a fly, if it would sit somewhere and it gets a looming stimulus, so that means kind of a disk that gets larger, it would very, very quickly take off and fly away. So that's also kind of motion information that is used for that. Mm-hmm. But it's interesting that this kind of wide field motion, where a huge circuit is dedicated to it, it's not as kind of obvious what it's used for as we would think it would be.
Yeah. Thank you.
So, any other questions?
Yeah, I think Sam has a question.
Yes, Sam, please.
Sure. Yeah.
Okay, so my question is related to future directions for this research. So I'm curious about how you decided on the Drosophila, the specific species that you're using in order to study this, and also what sort of next steps there might be in terms of the next possibly like animals that are or not animals, but organisms that maybe push this research in the direction of human. And
I'm sorry. So I don't understand you very well. Maybe somebody can repeat the question, or
Can you hear me a little bit better now?
yeah, a lot better.
Sorry. So my question is just related to future directions for this research. So you decided on the Drosophila. I can't remember the specific name, but I know that eventually you want to translate this to humans. So what sort of like are there other organisms that you are focused on maybe next steps, or if not, then like how did you decide on the Drosophila specifically?
So actually it all goes in parallel. So there's a lot of research being done in all the steps in between. So like as Chris also mentioned in zebrafish, so the cool thing about zebrafish is that they're also experimentally quite accessible, but they are vertebrates, so they have more similar eyes than ours. And there's a lot of research being done there. There's a lot of research being done in mice, I think in ferrets. There used to be a lot of research done in cats. There's also some in, actually now, I know that the lab of Botan Droska in Basel, they also do research on human retinas, which is kind of, as you said, ultimately, of course, if we want to understand how our brain works, it's important to study human retinas. So what they actually do is that they get the retinas from like people that have died in an accident, for example. But it's very, very difficult to do experiments in human retinas because apparently they degrade incredibly fast. So I think until now there's only a handful of recordings from human retinas because it's very, very hard to record from them. But at the same time, the circuit is kind of easier in Drosophila. So the idea is that you kind of understand how it works in Drosophila, and then you work your way up to more complicated subjects. But for me, did you mean for me personally, if I want to move on to a different model organism or to a different thing, or was that for the field in general? Or?
Oh, no, I'm just curious about how this research ends up then translating, not just translating, but integrating with other research on the topic, and how you go from studying motion and vision in this organism and then trying to make connections to other organisms.
Yeah. So, but that's kind of a general question about basic research. So I think the idea is not necessarily that kind of things from Drosophila have to end up in something applied for, I don't know, medicine, I don't know. But in general, like when you learn something in Drosophila, it's very, very often people later then find out that something similar can be found in other higher organisms, like in mice or in vertebrates in general. So actually the knowledge can often kind of translate between species or even between flies and humans.
Okay, so thanks again, Matthias. I hope you appreciated the topic as well, because for me it kind of reminds me on how evolution did tiny ML for a specific task. Obviously, the fly is a master of detecting motion, and so the circuit organization is definitely kind of well equipped to do that task, and that's the similar thing that we're trying to do in the summer school. So I want to close now. If you have any more questions, you can still stick around. Matthias and I will stay here if you have any more questions. And thanks again, Matthias. Really nice.
Thank you.
Thanks a lot.
Hello, Matthias.
Yes. Hi. Hi, Tim.
Thank you. I had to take care of something, so I'm not in the lab. I was listening to it from a taxi cab. But I was just wondering if you could take a step back and in your line of work with combining calcium imaging and electrophysiology, what's the technology you wish existed that could